how a pregnancy test frog helped unleash a deadly fungus. For decades, one widely used pregnancy test was not a plastic stick. It was alive. It had eyes. It had skin. It had legs. It was a frog. A woman's urine would be injected into the frog, and if she was pregnant, the frog could lay eggs. At the time, this looked like progress. It was faster than older methods. It was useful, and it helped doctors confirm pregnancies long before modern home tests existed. But hidden inside this strange chapter of medical history was another story, a story nobody could see at the time, because the frog that helped humans detect life may also have helped spread one of the most destructive wildlife diseases ever recorded, a fungus that attacked amphibians through their skin, a disease that moved silently through ponds, forests, and mountain streams, and a pathogen now linked to some of the worst amphibian declines in recorded history. This is the story of the African clawed frog, and how a pregnancy test may have helped carry a deadly fungus around the world. The African clawed frog does not look like the kind of animal that would change history. It has a flat body, smooth skin, powerful back legs, and small claw-like tips on its hind feet. That is where it gets its name. But the thing that made this frog important was not how it looked. It was how its body reacted to hormones. In the early 20th century, doctors and scientists were trying to solve a problem. They needed a reliable way to detect pregnancy. Today, modern pregnancy tests detect a hormone called HCG. But before small home tests existed, finding that hormone was much harder. Then scientists discovered something unusual. If urine from a pregnant woman was injected into a female African clawed frog, the hormones in the urine could trigger the frog to ovulate. In simple terms, the frog could lay eggs. That made the frog a living pregnancy test. No surgery was needed. The frog could survive the test, and compared to older methods, it was practical enough to be used widely. So African clawed frogs began moving. They were exported from Africa. They were shipped to hospitals. They were used in laboratories. They became part of medical systems in different countries. From the 1930s through the middle of the 20th century, these frogs were no longer just animals in ponds. They were medical tools. And for a while, that seemed like progress. But progress sometimes carries things nobody can see. Because some of these frogs may have carried a hidden passenger, a fungus called Batrachochitrium dendrobatitis. Scientists usually call it BD. BD causes a disease called chytridiomycosis. And what makes this disease so dangerous is that amphibians are not built like mammals. Frogs do not just use their skin as a covering. Their skin helps them breathe. It helps them absorb water. It helps regulate important salts and minerals inside their bodies. So when BD infects the skin, it is not attacking something minor. It is attacking one of the systems frogs depend on to stay alive. As the infection gets worse, the skin stops working properly. The frog can lose control of key electrolytes like sodium and potassium. And when that balance collapses, the heart can fail. That is why this fungus is so devastating. It does not need claws. It does not need teeth. It does not need to chase anything. It can kill through the skin. But the African clawed frog appears to have had one terrifying advantage. It could carry BD without always dying from it. And that matters, because the most dangerous carrier is not always the animal that looks sick. Sometimes the most dangerous carrier is the one that survives long enough to travel. And African clawed frogs traveled. They moved through medical trade. They moved through laboratory research. They were kept in colonies. And when newer pregnancy tests replaced them, some were released or escaped into the wild. In some places, they became feral. That is where this story becomes bigger than medicine. Because once a carrier species enters the wild, it can become a bridge between a pathogen and animals that have never faced it before. Native frogs in other parts of the world did not necessarily have the same relationship with BD. They had not evolved under the same pressure. They did not all have the same defenses. So, when BD reached vulnerable amphibian populations, the effect could be catastrophic. In some places, frogs disappeared before scientists even fully understood what was killing them. Streams that once had calls at night became quiet. Species that had survived for thousands, or even millions of years, began vanishing in a human lifetime. And the worst part was how invisible it looked at first. This was not a forest being cut down in one day. It was not a fire. It was not a predator people could see. It was a microscopic fungus moving through ecosystems. By the time scientists identified BD as a major cause of amphibian disease, the damage was already massive. Amphibians around the world had been declining for many reasons: habitat loss, climate change, pollution, invasive species, and disease. But BD became one of the most frightening pieces of that crisis. Because it could move through water, it could persist in environments, and it could strike species that had no obvious defense. This is why the African clawed frog became so important to the investigation. Scientists began looking backward. They studied preserved specimens. They tested old frogs from museum collections. They searched for traces of B in African clawed frogs collected decades earlier. And the evidence pointed to something disturbing. Scientists found evidence of BD in African clawed frog specimens collected decades before the modern crisis became obvious. Researchers also found BD in introduced feral African clawed frogs in places like California. Now, that does not mean every outbreak came from one frog. It does not mean the African clawed frog was the only way BD spread. But it does support a dangerous possibility. The global movement of these frogs may have helped move the fungus, too. And that is the real horror of this story. The pregnancy test itself was not evil. The doctors using it were not trying to destroy ecosystems. The frog was not a villain. But a useful animal was moved around the world without anyone fully understanding what it might be carrying. That is how some ecological disasters begin. Not with a monster, not with a plan, but with a small blind spot, repeated thousands of times. A box shipped here, a lab colony moved there, an animal released after it was no longer useful, a pathogen carried quietly into a place where it did not belong. For humans, the African clawed frog was a tool. For BD, the global movement of these frogs may have become transportation, and for vulnerable amphibians, that movement may have been deadly. The tragedy is that frogs are not minor animals in an ecosystem. They eat insects. They feed birds, snakes, fish, and mammals. Their tadpoles shape aquatic environments. And because their skin is so sensitive, amphibians often reveal environmental problems before many other animals do. When frogs disappear, something larger is usually wrong. So this story is not only about one frog, it is about what happens when humans move living things faster than nature can respond. It is about the hidden cost of biological trade. And it is about how a medical breakthrough can have consequences nobody sees until decades later. Today, African clawed frogs are still important in science. They have helped researchers understand development, genetics, and biology. But their history also carries a warning. A species can be useful and dangerous at the same time. A breakthrough can solve one problem while creating another. And sometimes the most destructive events in nature begin with something that once looked harmless. A frog, a test, a shipment, a fungus nobody noticed. And then, silence in the places where frogs used to sing.